Announcements. First of all, a reminder that for all the lectures you have attended, especially the guest lectures, it is possible to suggest exam questions which might, if the question is of sufficient quality to be used, net you bonus points on your final grade for the course. Not many people have used this option yet, but if you do have questions, make sure to upload them. Second thing is, as of next week and the week thereafter, the first hour slots for the lectures is not scheduled, which means that those hours are free. So tomorrow on Monday, next week on Monday and Tuesday, and the week after on Monday and Tuesday, we start an hour later than we have thus far. Right, you had a question.
What's the deadline for sending in questions?
That is a good question.
We haven't said it, right?
I seem to recall that we posted that in the Canvas announcement. If not, I'm going to spitball it at the day after the last guest lecture. Any other questions? All clear? Excellent. Then today we have with us Jose. You've all met him before, I'm fairly certain. He will be talking to us about the convolutional neural networks. And then a paper by Kelly et al. Which applies that towards finding sequence motifs, de novo. Hope you've all read the paper. It's a good read and it will make following the lecture much easier. So without further ado, Jose, the floor is yours.
Okay. Hello. First lecture ever, so let's see how this goes. So today I'm actually going to be giving two lectures. The first one is an introductory lecture, this one, on convolutional neural networks, and the second part would be, as Bas said, the discussion about the paper. Last year we didn't have this lecture, so everything's from scratch, and I have made it shorter by design because I'm expecting it to be question intensive. So the concepts are not necessarily the hardest, like the maths behind it are not the hardest, but I think the way of abstraction that you reach with this might require you to stop me and maybe ask some questions. So I have accounted for that, so don't be afraid, just ask in the middle of the session. So with that, I'm going to be talking about convolutional neural networks, but I would like to start with a picture. And this is a picture taken in 1911, and this is one of the first pictures taken in color. You might be asking where the color is, of course. Of course, by then, it's not the very first one, but they were very scarce. And the way of obtaining a picture in color was by simply taking 3 pictures with 3 different color filters, and that way you were blinding yourself out of some of the information. And with that, you were obtaining 3 different pictures here, and by working together these pictures that were partially incomplete, you could reconstruct and have a picture in color. Why am I talking about color photography? Because the colors were there. This was a property that was already in the image, but you couldn't get it with the technology that was available. You could only see light, yes or no, different gray of course, but black and white. But applying these filters, blinding yourself, you enabled some of these colors to be captured, and you would obtain a deeper understanding of this image by applying these filters, by removing some of the information and looking at the picture from different perspectives. Why did I choose the image analogy? Because convolutional neural networks are used a lot also for image recognition. You can use it for face recognition, for finding order, and there's many other applications like finding whether a vehicle is a car, if it's a truck, if it's a truck carrying trucks, I guess you could do that. But that's why most of the examples are going to start with images. But before that, we're going to start with an overview of the convolutional neural networks. We're going to be decomposing this image and butchering it and looking at the different spots. But this is the general, one example, a very general example of what a convolutional neural network would look like. You have 2 phases. One of them is feature learning, and the other one is specification. And what makes a convolutional neural network different from a normal neural network is the feature learning step. And that's what we're going to start with right now. So, So, in short, what you're doing with feature learning is you're looking at you're letting the computer learn what are the features of this. So, for instance, if you weren't doing this and you want to, let's come with an example. If you want to interpret a traffic jam, maybe you would have to say there's this many cars in here, or the image doesn't have a blur because of the speed, or many other things. If you let the convolutional neural network filter itself out with some of the filters that we were talking before, just blinding itself from one way, it starts detecting some of the features, some of the patterns, and maybe can recognize some cars and eventually taught the image as traffic jam, yes or no. The way you do this, you blind yourself via kernels, which are the equivalent of filters. Filters can also be used, but usually kernels are similar. As I said, these kernels are extracting different kind of information. One of them could be border, or vertical border, horizontal border. We're going to see some examples of that I have created with some of our pictures. It's in convolutional neural networks, which you're going to see refer to CNNs. That's how it all starts. So we're going to start with the convolution. This is the part of filtering. This is the filters again, because this is the most important thing of it. As you see here in this example, you've got an image of a car, and from this you're looking at it through different filters, and eventually here you see a matrix really that has a depth of as many filters as you have used. Now we're going to look at how these filters work. This is a car. This is the input. This is not the car. I haven't made the image of the car. But this is a representation of the car. And this one, the filters. If I slide this kernel as a window through here, I start multiplying all the values of the kernel with all the values of the input. And then I make the sum of the products and I obtain this value. One single value for each of the sliding events I have made there. Let's see this applied. This is a picture of our Christmas movie night, I think. Yes. And just for the sake of simplicity, I have turned it into black and white. But this is not an image anymore. This is a plot. I have detected how much light was in each of the pixels in this image. I have turned it into a heat map, and this is grayscale. Okay, first sanity check, we've got it there. There's an image. So now, I got this image, and I created two kernels. And this kernel is going to detect the vertical borders, and this one is detecting the horizontal borders. Can somebody tell me why? How could you see this from only the numbers?
Because the display of the numbers.
Sir?
Because the display of the numbers, the -2s are in a row,
yeah.
-2s are in a column, depends on it.
Okay, so if we're looking, for instance, let's see something here. This is pretty vertical, right? And they should appear, yes, it appears there also. You're seeing a border, at some point it has a darker color and another one it has a lighter color, right? So lighter color, if we're only looking at intensity of light, is going to mean higher number, right? Let's go back to here. So if I am using negative numbers in here and positive numbers here, can you see how what's going to happen here? If I have so let's say in this side I have a higher number, so it means it's lighter, it's brighter. Let's say I've got ones. Here I've got 0.5 in all of them, and here I have a 0. And now I multiply it by -2, 0, and 2. I'm going to have a negative number, right? Yeah, this is the part why I wanted to go with that. So, if I'm multiplying here exactly by the same, and I've got here it's a darker, here it's a lighter, I'm going to see represented on the one side it's going to be lighter because simply the product is making a positive number. Another one is going to have a darker color because the numbers have the sum of the products is negative. But now we're looking at here. There's nothing happening here. Let's say that all the numbers are the same. You have 0.5s in all the 5x5 grid that you're going to be multiplying from the input. What is happening if I multiply by this? What is the sum of the products? 0. So you see no feature here. There's nothing learned. So there's nothing learned yet, but at least there's nothing extracted. There's no edge there, there's no anything. I chose this image also because this other one is designed to extract horizontal ones. And there was like this very nice horizontal line here, which appears here, all of them, and not here. So that is how an image has, like this CNN, the convolutional neural network, in the first steps, has gone from just having an image, just having different intensities, brightness and intensities in the peaks, to have an actual defined border. So that has happened. This was the first step here. And now something that is done, I think in all the CNNs I have seen, is pooling. And pooling, as you can see here, is reducing how big this matrix is. And the way you do this, pooling and subsampling are used equivalently in the field. What you're trying to do is extract as much information from the product of the kernel and input that I showed you from the convolution step into a smaller representation of it. Smaller representation that still keeps the gist, so the most important part of the image. So you're moving here, you're reducing by, well, in each of the dimensions you're reducing by half how many, like the size of the image. And here you can still see, if this is a sign that starts with 60, here you can see 16 here, you can still hint it. Maybe it's not as much as you saw it before as a human, but this will allow you to find other kind of 60s. Mathematically, what pooling is doing is dividing the image in different regions, and then you've got many ways of doing this. The most popular one is max pooling. So what you do is you check, for instance, in the orange one, which one is the highest value. So you take that one, it's 255, and you do it for all of them. And that way you end up either, so you obtain that. But there's also another one called average pooling. I want just to show you that although the max is the most popular one, it's not the only one. It has, all of them have drawbacks. If you've got an input image like this, which has like positive values as white and like zeros as black, if you're getting the max, you're completely losing. With the max pooling, you're losing all the signal there. If you're going for an average, you still have it there. If you're going for something that's the other way around, you've got the like everything is zeros, and then you've got a diagonal that is all made of ones. Well, 255s, I don't care, same. If you're going for max, you're retaining all the information in a smaller size, but you're retaining all the information. You can still redo the same shape you had before. You're doing for average, you can still keep it. It's lighter, it's not as intense. So what I'm trying to say is like both of them have drawbacks, but also the most popular one so far is max pooling. So that's what you're going to be finding most of it. Yes?
Yeah. Isn't it with the top one that you're just putting emphasis on the surroundings, which is why they have the maximum value, right? With the bottom one, you're putting the emphasis on the bar itself.
Yeah.
So that's why you see after max pooling, you will see the bar, and with the top one, you're actually, you just focus on the surroundings instead of just the black bar.
Well, you're not focusing on surroundings in any of the cases. This is purely numerical. You're going for like maximum, and this is a very, I think, intuitive case for us because we can just see a line, and later on you're going to be applying it to way more complex problems, of course. But what I was just trying to show here that it's like maybe simply because of the problem you're using and you don't know it in the first place, you might be having different effects of the pooling. Just it has been tested in many cases, and usually the one that works the best is max pooling. Maybe for some situations, and somebody here is an expert on that, they tell me no, average works better for this one. Could be. I don't know. I just know that from all the CNNs I've been reading, they have checked that the max pooling is the one working the best. But yeah, well, why pooling? If I'm explaining pooling, first of all, you're reducing dimensionality. We're talking that usually the problems you're applying these networks to are either very complex or you have huge data sets, millions even sometimes. Everything you can reduce is going to be good for your model. It's going to be easier to train, it's going to be faster, but that's not the only thing. You're also improving the generalization. If you remember the picture of the signs, you could still capture the gist of every filter, like what the filter was getting, with a smaller representation. So this prevents, or can prevent overfitting, but it also eases the classification of differently oriented objects. So for instance, if you have been training for cars and you're showing a car that's larger or smaller or is twisted, there's different ways of pulling. It doesn't have to be only in the same layer. I'm not going to dig into that, but it would enable you to find a better fit, basically, so to actually detect it. Sorry. Do you want to know more about why a car twisted would be detected, or is that something that we want to? Yeah, see, yes, yeah. Okay. That was too much. Okay, so far the one I have shown you is you've got like only one. Everybody can see it because this is in between. Yeah. Okay. So let's say that you've got from the initial car. Okay. I'm gonna do it with a 6. It's easier, and I've seen this example online. You've got a 6, and you filter it, and you find only like the edges. So from here, you do like you multiply by kernel. This is the input, and kernel whatever, and then you end up with let's say only the borders of the 6, something like this. Yeah, I'm making this up, by the way. Okay, so you end up with this filter here, and then what I'm telling you is like if I reduce it, I maybe have exactly the same but smaller, but let's say that I capture all of it. This is still kind of a 6-ish. Okay. Now this is like the training of it. This is the training image I have put. But now I'm like, I want to know if I have a 6 in something that I'm finding. But the 6, the image of the 6 I'm giving is this. The other way. 6. Okay? So now what is happening is, what happens if for some of these filters, instead of obtaining this one, I obtain, I'm going to twist myself to make it better. I obtain the 6, and for another one, I obtain the other one here. Okay, so this, if I outline it as like normal 6 going up, this is twisted 6, twisted 6. Okay, so now, instead of pulling only from one of this, I'm going to make what it's called like, I think it's called multi-layer pulling, or yes, multi-layer or multi-filter, can't remember right now. So, and I make a max pulling. Do you remember the max was like, out of these options that I have, I'm going to pick only the maximum. This one would match with this one, and would be recognized as a 6. This one would not match so well with neither of these two. So maybe it's assigned as a 9. Because it's twisted. I don't know. Maybe the 6 wasn't the best either. But if this would allow to give an almost perfect match simply from having a different orientation. They're specialized CNNs on this. So like this is like the very basic of it. And maybe we should go more into that. But what you should remember is that it helps generalization, it prevents overfitting simply by reducing it, and it makes your classifier more flexible. That's best from pulling. Something that Bas told me it would be a good idea to already introduce in this lecture is, I'm talking about images, but the paper was about sequences. So you're like, how do I work with this different dimension? So, basically, how do we do exactly the same thing, but instead of with images, with the sequence? So, there's this one thing called one-hot encoding. You've got your sequence, and basically what you do is you transform the image into a heat map and only give like a 1 to this position of the sequence that you're in, and a 0 for the letter that you have in the position of the sequence, and a 0 for the other ones. That way you've got a representation that looks a bit more like an image already, right? Then you can basically operate the same way. You've got a kernel, you've got different values for it, with which when you multiply with this, you would obtain a radial value. So this is exactly the same thing you were doing here, just with one dimension. Keep in mind this would be sliding through all the sequence here. Okay? This is the part, well, this was the end of like the convolution part. And I'm almost sure that there might be a question about this still. And we can go back later because we come back into this into the next lecture. But if you've got questions now about it, please ask. Yes?
Does it matter the way in which if you come back to the previous picture, the image cycle?
Yes?
Yeah, instead of arranging ACGT, you choose another criteria, for instance, saying the whole sequence, or instead of, if you have a long sequence of DNA, creating a picture that fits the dimension of that array. So my question is, does it matter for this purpose to choose a different setting of the image that you're going to generate?
You mean if instead of ACGT, it's ATGC?
No.
No?
I mean, like, if you have, I don't know, 1,000 sequence long,
Okay.
then you can maybe create an image of 500 by 500.
Why by 500?
Well, sorry, 10 by, if it's 1,000, 10
4.
by,
Yeah, but that's what,
Like it does not matter.
that is what it's done in the paper. And Basset, they encode this, like in this case, you only have this one's here, and then you can make it longer. Is that your question? Because I'm really not following.
The question is, does it make a difference if you arrange the ACGT instead of arranging the image in another array? So if there's a difference in the size of the way of the picture is represented.
I am not getting your question. I don't know if anyone is, or if you can translate me this. Sorry. Sorry.
I think you can increase the size.
Yeah, of course. Yeah.
You can increase it vertically. And
Yeah.
it
I
probably
mean,
matters.
or you can just plot this horizontally. That's also fine.
It probably matters if you change the size vertically, right?
I'm asking this because we tried this, so
Yeah.
but it didn't work so well for us, so maybe we missed something. So
This is just a representation of the sequence. Instead of putting a letter, you just make it numerically. You make it.
because at the end you're gonna train your neural network on images, so to say.
Yeah.
So
If I can pitch in on that,
Yeah.
it might be useful to think of this less as an image
Yeah.
and more as a matrix.
Yeah.
So you see on the right-hand side of this slide, right, that it's not an image, it's a matrix of zeros and ones. Same with the complete left-hand side of this. Now it looks like sort of a dodgy QR code, right, with black and white blocks. But it's actually a matrix of zeros and ones. So you can encode any length n sequence in an n by 4 matrix. 4 because you have 4 different nucleotides, so every row, or in this case every column of that matrix corresponds to one of the nucleotides. If you have a 1 in that position, then that nucleotide is in that position. And if you have a 0, then it's not that nucleotide for that position.
So how
So
would
suppose you were to use a length 600 sequence, as they do in the Bassett paper. Then you would have a 600 by 4 matrix of zeros and ones representing 600 A's, T's, C's, and G's. Does that answer your question?
no, okay, maybe.
Then maybe we should take this up over the
maybe
coffee.
we made a mistake because
So
what
how-
we
how-
were trying to find out is generating pictures of sequences, and then from those sequences try to find the regions. We wanted to find important characteristics, so like trying to reverse engineer the neural network to find like reaches or particular in the first layers of convolution, try to find the regions that are important, but we couldn't do it.
I think this is better over coffee because it's a bit more of that.
Okay.
But like, since we're talking about matrices, how would you represent a protein sequence then? Someone? So using this one-hot encoding, how would you represent?
10 by 10.
Yeah, and by 20. Perfect. That's it. So you get the one-hot encoding if you can answer that. Perfect. But yeah, basically it's exactly the same principle. You're making input by kernel times kernel, and then you get that. So now we're going to yes.
What if the data is not, I mean, you cannot transform all the features into like one coding coding? For example, you have sequence, okay,
Yeah.
but also numbers or other values.
So which kind of features, for instance, hydrophobicity?
In my know, let's take the actual assignment. We have starting point to G, ending point to G, and then a lot of.
But then you're not using, you're not thinking about sequences anymore. You're just talking about
No,
features.
yeah,
You're not learning, like you're not letting the neural network learn itself about the features. And the assignment you were already given, this is how much it has doubled or it has been reduced. So like then there's no learning from it. Like there's no learning features. It's just training the network.
Yeah, the thing is, I'm not intended to apply to the assignment.
Okay, okay.
Just what happens if the data cannot be transformed, like all the data cannot be transformed to one hot encoding?
Well,
have other values.
You don't have to use one hot encoding. Like one hot encoding is a way of using like actual sequence data. But if you're dealing with something that comes from the sequences and like like there's a duplication or there's there's a methylation spot somewhere, like you can already fit that into position or feature yes no or 0 1 2 or -1 and like there's. So my point, like this part was about learning from an input that you don't have to describe. Okay. So if you have other features, you can use them, but it's just differently. Yeah.
Yeah, I have a question about your example you do on Blackboard.
Oh yeah, sure.
Did you mention possible layer pooling?
Yeah. Well, there's like the pooling that is explained here is the one I made like. So the pooling I'm doing here would be this one, but there's another kind of pooling which is the one that goes through different layers, and that one is the one that gives you the flexibility of the car. I asked you about whether or not you wanted to know it because it's a bit different how it's used on Basset.
Yeah, for me it wasn't clear with them.
Yeah. So if
one step through all the layers or did all the layers?
if you want, so this is the way that pooling is used in Basset. Okay. This would be the closest to it. Like you've got a like a product of input times kernel, and you want to capture as much of this output in a smaller representation, a small dimension layer, so input. Well, future input, of course. Yeah. This is the way that it's done in Basset, but I'm trying to give a broader overview here. Okay, so is anyone ready to go to the next one or not yet? Yes? Okay, good. So basically what you have done with this, like you have allowed the computer with neural network to just extract the features, to know what is in there in its way. But then you still have to feed it into a neural network for a classification or for learning things from out of that. So I want to make a disclaimer. I want no one to say in the exam that a neural network is an artificial brain. Okay? It's recorded. Okay? Don't. It's recorded, right? Good.
Of course.
Okay. It resembles that, and that's why, especially for bioinformatics, I think there's many people that come from a biological background, so I think it's good to just see the similarity between the neurons in an artificial neural network and a neuron in our brain. So you basically have inputs, which in the brain are dendrites. Something is happening in the body, right? So you make either the sum of the signals, and you have a trigger in which at some point something's happening. Then you've got an output, which is the axon, and the axon triggers if the axon is not the output. This is exactly how artificial neural networks are organized. Each of the circles you see here are a neuron. Well, yeah. So, this one's here actually. So, what you're doing, let's look at this one here. I think it's in the next slide, it's better. We take only one. Then you've got here 3 inputs plus a bias, which is kind of an input, and I should have put a WB here, the weight of the bias. Each of these inputs has a weight. You remember weights from probably many courses already. It's how much importance you're giving to the signal that's going on. You make the sum of all these signals going in, and you obtain the summation of it, and then there's an activation function. This activation function is going to determine, with all this input that I'm having here, all the signals that are coming in, how am I processing it? Here they come. Basically, you've got so the x here in any of the signals will be how much is going in. What is the addition of all the signals that are going in? If you use, let's go for the sig mode for instance, and the signal is 0, your output is 0.5. Well, you know how to read plots. This is the one that's used the most, like relevant. This one here, what it's doing is it's removing all negative signals. So if you've got like the summation of all the signal times product going in is negative, you're gonna just give 0 for everything. The rest is just if you get a 1, it's a 1. If you get a 2, it's a 2. This is the one that's used the most. This is right now applied to neural networks. I want to maybe make a small disclaimer or maybe addition, but you can also use it on the images we were looking at before. Many times they don't want to have the negative values and they just want to have the positive ones, and you can see they transform the image or the input so all these values are removed and you increase nonlinearity, which usually which improves the performance of the neural network. So you've got many different kinds of this. Some of them have drawbacks, some of them, well, all of them have some drawbacks, of course. The reason why this one is the most used, ReLU is the most used, is because it reduces the chance of having vanishing gradient, which is something that I put at the end in case you want to expand. I'm not going to go into that, because otherwise the lecture gets too long. But basically, using these functions, instead of just directly mapping it one-to-one to what was coming in, it's exactly what's coming out. What it's doing is allowing you to capture behaviors that go beyond nonlinearity. And, well, I guess most of you, at least everybody that is in the Masters, has done systems biology. So you have seen how many elements with nonlinear behavior, so enzymes, if you put them together, can show some emerging behaviors. Frank is really keen in this thing, so I'm pretty sure he showed quite some of this. That's simply because of the nonlinearity. If you start only putting linear effects, these effects become very boring. So you basically can only get flat or slope or not much more than that. So that is how an individual neuron works. But of course, you want to obtain something out of it. So you want to train your neural network the same way you train any model, right? So the way you train it, simply by minimizing the cost. What is the cost? The cost is like the output you obtain from your neural network minus the target, which is like what you would like to have ideally. Okay, so let's say, let's go back to cars and trucks. A is car, yes or no. B is car, yes or no, or probability there. You know that the first one is a car, but then it doesn't give you a car. But which things can you tweak? You cannot change your inputs because they are what they are. They come from before. You can change your weights. You can do it because that's just the importance you're giving. And you can change the biases, all of them. So now basically what you want to do is, if I have a 0.3 here and a 0.7 here, I want to change the weights for first neuron A in order to increase it, and neuron B to decrease it. And every time I will get closer to this. You do this for every training instance.
What do you mean with bias? Like random stuff?
Bias is just something you can add. So you've got all the inputs that actually come from previous neurons, and you can just have a value that you add simply because it works better. So that's something you can have. I think someone you do neuroscience, right? I think no. Someone doing neuroscience, I think that also happens in neurons, right? You can just have a normal build-up of. You don't need a signal in order to contribute for the trigger, if I remember correctly. You can just have it internally from the neuron. So, but yeah, let's not make more analogies maybe with the actual neuron. Yeah?
You may change the bias, but isn't the bias usually 1?
No, the bias would be just a number that you could, the same way that you've got, oh, this is also here, you've got input from neuron 1, 2, 3 in the previous layer. You could also have another thing that doesn't depend on the previous layer, just adding in. Okay. There's also like, there's other network topologies that make everything a bit more complex, and you could even have some input from even earlier neurons going in, but that's beyond the scope of this presentation. This is simply like, this is how a neuron works. So, basically, things you can change. Weights, here, and biases, here. You can change the parameter for each neuron, so it approximates more to the target. And you want to do this in a way that it makes all the training instances get closer to the value they want, not only for one. We're going to expand a bit more on that in the next one, because now we will start with the concept of batch versus mini batch, and yeah. So yeah, basically you want to minimize the difference, you want to minimize the cost. You start from the output neurons and all the way back. And yeah, and this is called backpropagation. This is explained a bit in more detail in the next presentation. But I think it's basically the chain rule, but implemented in a very efficient way. I said I was going to go back to this because I was talking about the training instances. Well, I talked before that neural networks are usually used for problems that have a lot of data. This, you could use all the data for training. That could be done, and I don't know how many years it would take. I probably not much, but it would take a long time. And in that case, you would be just using, like, iterating over all the sequences all in one step, and you would be having a normal gradient descent. And I have added here something. You see, number of iterations equals number of epochs. This makes more sense with this. But like, what you do, like right now, you remember from hidden Markov models when you were training BAMWorld, you were iterating all the time. So when you were iterating through all the sequences, I think they had like 100 sequences. You had 100 sequences. That was one iteration. And then you were starting again. Then you were starting again. That was one iteration every time. What happens if you divide your data? Instead of this 100, you do 25, 25, 25, 25. Then you just stop at some point. Every time, every division that you would make there would be called a mini batch in neural networks. Every time you finish one of these divisions, you would have one iteration done. Once you have gone through all your training instances, all your data, then it becomes an epoch. I'm saying this to build up vocabulary because this is the way you find it in papers if you want to understand it. Yes?
What is epoch?
Epoch is, one epoch is one pass through all the training instances. Whereas one iteration is one training with one set, which could be a mini batch.
How is the data split into mini batch?
The what?
How is it split into mini batch?
Oh yeah, it should be done as randomly as possible, but I think there's also some writing about it, like, yeah. Yes. So, what you're doing if you're dividing it in mini batches is you're effectively doing a stochastic gradient descent instead of an actual gradient descent. Well, this is a picture of a normal gradient descent, of course, but there's also another thing when you're training, which is, I told you, you're tweaking the weights, you're tweaking the biases, but by tweaking, it means you're changing these values. How fast you're changing them is represented by the learning rate. If you are moving from, let's say, you've got, in the previous example, I'll go back here. Let's say here you've got a 0.3 and here 0.7, you want to have it reversed. If you go already to a 0.9 and a 0.0 here, that's going to be a very fast learning rate. And in this case, it looks like it works. Then maybe in other cases it doesn't, and you end up trapped in a local minimum. Here. Okay? Simply because you find it there. So if you're having smaller steps, you're moving slowly and you can detect easily how to get to a better place, and you're going to prevent overfitting. but it's also taking much longer. And with such number, like with all the amount of data that you've got, you might end up with computational time, like running out of time in the servers that many other people have to use. So this is more a practical issue. But then you want to, of course, once you have trained all your neural network, you want to put it to use. You want to predict. You want to predict what's coming out of it. So for that, you would include like a 6 or a car or a sequence, and you would like to tell you whether it's from one class, it's from another class, because you have training for that. This is a very fast problem. Like this is very fast to calculate. The training is very expensive because all this changing and tweaking of weights and biases takes a lot of time to train. In this case, you just put it in, makes all the calculations, it's out, I mean, relatively fast, of course. And well, this is the end of the introduction. Like, we're gonna go in more depth and a bit more fast in the next one. I have put here some more concepts that you might want to look at if you want to expand into neural networks. They are not, you're not restricted to this, of course, but I have found that it helps me visualize the papers and the architectures better if I understand them. And I think that's the end of the presentation, the intro presentation. Yes. We're going to go in more detail in the next one about the paper. But which questions do you have now? Because it would really help if you ask your questions now. Yes?
Yes, about cooling. Is
Yeah.
cooling applied on every filter, all the filters?
Yeah, you can apply it. So there's two ways of doing it. The way it's applied in Basel is on every filter. So if you look at the initial image through 10 kernels, you obtain 10 outputs, and then you pull each of them. But there's also an alternative way of pulling, which I think I just made you more confused by making the example of the 6 than by not pulling it, which is like I look at different outputs of these filters, and I just check which one is the one with higher value. And you can make it like pixel by pixel even, or you can make it all of it. There's different ways of doing this, but like the one that you're going to be seeing later on is the first one, the one which like you just take one filter and you reduce the dimensionality of it, and you expect you're going to obtain a good enough representation of the output with that reduced version.
Is there another filter then, if it is reduced?
Sorry?
Do we call it another filter after you reduce? You have the filter, then you reduce it, it's called cooling.
Yeah.
And then you obtain a new filter, or is
No, then you just have a matrix, right? You've got a matrix representing your data, and you can do more convolution steps there. There's many other things you can do. You can batch normalize it. Like that's just data that you have obtained at that point. It's a new matrix. Like I think this makes maybe a bit more sense. It's an abstract thing, but I think it might make more sense when you see it applied to an actual problem with BASIC. Yes. Huh? Anything Bas wants to tell me I was wrong in, or
No, I think we're fairly on point.
Good.
Perhaps we can test the students real quick. Suppose we have a 10 by 10 input matrix, right? We have a picture of something, we made it into grayscale, and now all the numbers are between 0 and, say, 255. And this is a 10 by 10 matrix of 100 numbers between that. And now to this 10 by 10 matrix, we apply a 5 by 5 convolution filter. So these kernels, for example, for horizontal and vertical edge detection that Jose showed in the start. What would be the size of the output of applying this 5 by 5 filter to our 10 by 10 input? Or in other words, how many unique positions can we find in the 10 by 10 input that this 5 by 5 filter fits on top of? Yes?
If you're using convolution, you also use a buffer, right, around the edge to get the same inner size.
There is a couple of ways of doing this, like you can repeat the edge values, you can wrap the filter, but in this case let's assume that we don't.
Yeah, it's like 4 with like no overlap.
Yes, there is 4 with no overlap, but the output of this would actually be 6 by 6.
Yeah.
So it's 10 minus 5 plus 1. So there is a 6 by 6 output for each time we apply a 5 by 5 filter to our 10 by 10 input. Suppose now that we don't just have the one kernel for, say, horizontal edge detection, but we also have a vertical and a diagonal. Right? So we have 3 filters we apply to our 10 by 10 input. How would you describe the dimensions of the output of these filters? Very good. Right, so we have 6 by 6 by 3 in the next layer. Now the 6 by 6 by 3 we can again apply filters to. These can be 2-dimensional, these can be 3-dimensional, these can't be 4-dimensional because we don't have a 4th dimension, but in the next layer they could be. But we can repeat this, right, across many layers. So we start very simple with 3 6 by 6 filters on this 10 by 10 input for, say, horizontal, vertical and diagonal lines. Right, now in the next layer we can combine our 6 by 6 by 3 outputs as the input for more complex filters. So we can start looking at shapes, for example. We've seen the image of what happens when you apply these simple filters to the speed sign, where you can go 60. Right? So if you only have horizontal, vertical and diagonal lines, then combining that you might start learning circles, or the way to, is it putting back up?
Yeah.
So it's a bit tricky to see, right? But in this row here, when you have applied your simple filters to the raw input, and then you've subsampled this to more parsable input, and then here again it explodes by applying simpler filters to this, or more complex filters to this, then you have a new set of features. Suppose we have our 6 by 6 outputs, and we subsample this 2 by 2. What is the dimension of an output thereof? Are you almost close to this? Right? Because the subsampling is non-overlapping. So if you have a 6 by 6 input for a 2 by 2 subsample, then the output is 3 by 3. Because there is 3 by 3 unique ways to place a 2 by 2 grid in a 6 by 6 box. Does everyone get that? Right. So in those steps, applying a 5 by 5 filter to a 10 by 10 matrix and subsampling 2 by 2, you've reduced your features to 3 by 3 by however many filters you started with. And this is important to understand because in the Bassett paper you will do something very similar. There you start with a 600 by 4 input matrix. This one hot encoded version of a 600 base pair sequence. And I don't want to mow too much grass away from Jose's feet, because he will be talking about Bassett as well in a second. But basically by applying simple filters to the one hot encoded sequences, which represent motifs, and then applying more filters to the motifs, you can find combinations of those DNA motifs. And if these were to represent, say, transcription factor binding sites, or the things that regulate DNA accessibility recognize, then combinations of those proteins binding to DNA might be very useful in predicting whether or not a certain piece of DNA is accessible in a certain tissue. And that is exactly why Bassett works. Because it starts simple from sequence, it reduces that sequence to a set of features representing what motifs are roughly in what position in this sequence, and then from combinations of those motifs and combinations of those combinations, you end up with enough information to predict if DNA is accessible. Now, if we were to do this beforehand, we would be lost, right? I don't know how many of you know what motifs proteins that regulate DNA accessibility recognize, and in which tissues those proteins are active, and in what combinations they have to bind to DNA to do something. But this is a very complex problem, and that is exactly why machine learning works so well for it. But yeah, keep thinking about what happens when you apply certain size filters to certain size input, and how that reduces or increases the dimensions of your data.
That's very important because, like, for the next lecture, if we want to actually go into depth into like all this filtering of finding patterns and everything, we will have to go a bit faster with this. So, yeah, this is what it is. You've got a lecture recorded, so you can check it later. But yeah, that's it. Let's have a break.
All right. Coffee.
Okay. So this is actually Sanna's lecture from last year. I think it was good. There's not need to change. There's some slides I'm going to go faster on them or even skip them simply because I have already gone through this initial presentation that wasn't there. Who has read the paper? So, okay, so like 20% or so. Well,
disappointing number of hands.
No.
Yeah, well, this presentation is made more like from the discussion point of view, so it's not, you're not going to get out of it as much as if all of you had read it, so that's your problem for the exam, I guess, but well. Okay, let's get you in this. Well, for those who did it, do you like the paper? No. No. Okay, did you understand the paper? Yes, no, something? No? So? Do you think you would have understood it better after this lecture? Yes. Okay, so maybe that's something for next year.
60%.
60% what?
Understanding better.
60%. Okay, well that's better than 0. That's good. Okay. Let's start with the the principle of what the problem is. So, for those who have a biological background, this is probably seen too often. But for those who maybe haven't explored so much, you want to know about the structure of DNA because you maybe only see it as a sequence, it's ATGC. But physically, DNA is folded and more folded. And it's at different levels, and these levels are regulated by histones and methylations and acetylations and many mechanisms that they are still studying. Otherwise, ask Jan Kotter. He's a lover of this. So, normal DNA, how you have it folded here, goes into a histone and then it folds already into the nucleosome. Then you can observe how many of these histones folding DNA makes the pattern beats on the string. This ones you've got another kind of histone that you can add, and then it folds even more, and that way you obtain the 30 nanometers fiber. Why is this relevant? If you have a protein that has to access the DNA, and the part of the DNA you want to access is like this, is it going to be able to reach it? No. You want to be able to access usually the big groove in here. So for that you need them folded. So this is a problem. This is something that's used for regulation of everything. So you have different genes activated when you're growing, and when you're already an adult. You have different genes if you're, well, not necessarily you, but like eukaryotes in general have different genes activated if they are in a high heat environment, cold, whatever. Different stimuli make the accession different. Also, you've got the same DNA in your liver than in your brain, for instance, yet you've got different expression. This is one of the mechanisms in which this is regulated. So, let's see this. Yeah, I said that already, and I think I said this already, so good. But then there's a way of checking accessibility of parts of the DNA, which is using DNase I hypersensitive sites. Basically, this is a mechanism used to find regulatory regions on the DNA, and it's based on if the DNA is unfolded. It's cleaving, if I remember correctly, right? Yes? it's cleaving, and then you can sequence it, and then you know that this part is accessible. You can do that over a cell line, and then you obtain that. So this is what you would obtain here. The DNA is binding to a region in which you don't have this histone, so everything's unfolded. So in this region of the genome, you would find signal, and then you know this part is open, therefore it could be a regulatory region. What you find is that in different cell lines, you've got different patterns. There's some of them that are always their household, so the regions that are in charge of regulating household genes are probably open all the time, or even the genes themselves. The regions that are only tissue specific are going to be only accessible in those tissues itself, but not in the other ones, or maybe in some of the other ones. I think we already, yeah, well, so there's always overlap, of course. So you can see here, this seems to be accessible in all the different cell lines that this study has. Whereas here you find this peak here. This one is only accessible in those two. So this is exactly what I was saying. For those who have read the paper, what do you think is the aim of this study, of this paper? Don't be shy. Yeah.
To create a model that can predict sites that are accessible.
Yeah. Well, that's one of them. Yeah. Something else? Yeah. I mean, that's what the model does, but there's more information extracted from the model that's very relevant, right? We'll go into that later if you don't know it now. So, basically, what you are having is like you are giving a sequence, you know the dimension of this sequence. You're giving sequences, you're inputting sequences into this model. It was mentioned in the paper that it was 600 base pairs long, and they were finding it from a database, and there was like a SNP that they knew that was relevant for whether or not it was accessible. That were taking the 600 base pairs that were surrounding them. So, what's the dimension of each of the sequences? Come on, we talked about this in the previous one. One sequence, what's the dimensions? 600 by 4. Okay, so you want to have as an output, she said, like you want to know whether a region is accessible or not. The extra thing is like you want it by cell type, and you do this by checking what is the sequence precisely for this cell type. No, that's good enough. Well, yeah, now let's go for this. Which kind of information, if you had to give an information to the classifier, which kind of information would you give it? Would you give to it? We're talking about sequences, and you know that there's some regions probably that are more accessible than others. Mm-hmm. So how would you, so which information would you give to, I want to find openness or non-openness in the DNA?
Like regions that we know that are usually accessible, like motifs?
Yeah, that could be a way of finding it. If you don't know anything, just let's say we make a Control F over the genome and we start pasting a sequence that we know. If you find it, that's likely to be a region that is open, right? Okay, good. Well, you can find here, this is a transcription factor here that binds to the DNA, and we know that it binds to this. And what we found from the paper is that Bassett predicted that this was one of the possible regions that would make DNA open and would make it accessible. There's some resemblance there, and this is extracted. This is one of the filters that, remember we were talking about the car, the first convolutional filter. There's one of the filters in the first convolutional layer, sorry, that is giving significant. It's showing that this region will be open, and this is detected by ADASIT after all the training. Just for you to know, this is some of extra output that you could obtain besides whether this sequence is accessible or not. You're actually getting some information about which things to look into, because this one is known, but there's others that are not.
Did you provide a filter that was looking for this sequence?
I can't remember if they actually did. I think it was randomly done, right? Yes. But you could have, like, that would be a nice experiment actually. If you know the filter, if you know the motive, you could do this a filter and see if it actually has, like, if it shows as significant or not. If it shows as something that is creating this discrimination. So that could be something you do, but they didn't do it. I think they made it just randomly. 600? No, how many filters? I can't remember. There were many.
I would say 10,000.
10,000.
It's like enormous.
Yeah.
And not all of them ended up being relevant for the final predictions. But the important message on this slide is that without telling BASIC anything about these motifs are important, it has found the bottom sequence logo. Has everyone seen those before? A motif logo like this?
Mm-hmm.
Nolly? Good.
There's a very nice plot layer. I don't know if I have it in the presentation, but I can show you in the paper, in which it shows the importance that is given to some of the motifs, and then they are colored differently depending on whether they were known to be motifs for this or not. That's a very nice thing, because it detected others that weren't even there. Okay, so CNNs. We have talked about this before. Who can guide me through this right now? It's not car now, it's a sequence. Come on, be brave, be brave. Even if you haven't read it. No? No? No. Okay, I've got a sequence, it's still the car, right? So, you remember the one hot sequence, you've got a matrix of 4 by 600, basically. So, this sequence is passed into the convolutional layers, and these are the filters that I have shown here. Okay, so you've got filters here, and what you're doing is you're checking how you're basically multiplying the one hot encoding, moving through it. You're convolving in one dimension, only moving in that direction, and then you're getting, for each of the 3 filters you've got in this example, you're getting how much you're getting out of it, what's the score you're getting out of this sum of products that you were getting. Then this is the actual output, so the blue, mostly blue thing. Do you remember the activation function I told you about the reload? That one was removing everything that was negative, and then it was keeping only the positive ones. Blue or negative, all of them gone. Okay? And then max pooling here, I think they just, there's no lines in between here. They just didn't make it narrower. But actually here, if this was, well, 300, this was 600 by 4, can't remember the length of, how?
19.
19, so it was that, 18, so, oh yeah. The math, we asked about that. What's the size of this one here? We start with 600 here, or here, same thing. We apply an 18 length, 19, yes, sorry, 19 length filter, and we end up with this. What's the dimension of that, of the output? Oh no, this is cooling, forget it, not this.
600 minus 19 plus 1.
Here.
Yeah.
Sorry?
Minus 600 minus 19 plus 1?
Yes. 600 minus 19 plus 1, which is 582. Yes, perfect. And then you remove negative ones, and then here you're pulling. I'm not going to do this by heart now, but how much, what's the dimension of this if you start with 582? Or which operation is it? Okay, you don't know actually how much I'm pulling. Let's say I'm pulling by 3.
divided by 3.
Yeah, that's
Yeah.
it. Okay, good. Sanity check. Good. We get that. Yeah?
Why 3 rows? I just can't make sense of that.
Three.
You start with
Oh,
4
it's 3
rows.
rows.
Yeah,
Okay.
there's 3 rows. And then
So, actually it's 10,000 rows.
okay, so it's not okay.
It's
yeah,
it for each of
that
the
makes
filters.
sense.
Yeah.
Okay.
So in this example, there's only 3, so they kept it 3. But that's it. So now you've got already, like after doing this several times, because you've got 3 layers, if I remember correctly, you go into the neural network. We were with that before, and we already have like the neurons and the weights and the biases, and you start training, and at the end you just get the predictions. You're getting your predictions, and you start tuning it, and you see how do I optimize this to obtain the best predictions of it. So you basically train the model. This is the convolutional part. This is the neural network, and this is the final step you make in the neural network, simply to know the probability of from 0 to 1, usually to know whether the prediction is more likely or less likely. Yeah?
I wasn't.
Sir?
It wasn't 10,000 filters.
No, it wasn't. Then, yeah.
300 filters, yeah. I'm not sure about when you go one slide back, the last one.
Here? The linear transformation
Yeah, each
signal?
of these sequences has been given a classification, I suppose. That was a little bit unclear with me.
Sorry, what do
Yeah,
you
so
mean?
it has to put it somewhere, either open or
Each of these sequences, according to the
are
weight.
open always.
Yeah, for each of them, according to the weight that the neural network, the weights and biases the neural network has in that moment,
Yeah.
has been given a prediction, and then you're checking how good this prediction is. You do the overall your training instances, and you want the best, like you want to find the moment, the weight in which you obtain the best result for the average, for all of them, or as much as possible. Of course, you still have your confusion table, and you're going to have some that are not well predicted, but that's the thing. And then you adjust the weights for the whole neural network so that you will obtain the best prediction of it, which gets closer to the target. So, well, this is the convolution part. I showed you already before in the car example. It was the part on the left. This is the neural network part. That was more on the right. This is like the very last step of the car, simply giving an actual probability. Yeah, well, this is another example of that, but with letters. So let's just keep it because it's exactly the same thing. More of the same, I think we have already shown this. Yeah, so because that was simply like what you're putting in, what you want to get out. You want to get out predictions, you want to get it as close as possible to that. Neural networks are probabilistic discriminant classifiers. What does it mean? Well, it's here. Like, who can maybe explain it in simpler words? Do you remember the hidden market models that you were actually interested in knowing the path of it? Okay, so that's a generative model. It's telling you how what you're having was generated. Here it doesn't care. It's just giving you the probability. So that's it. This is pretty much it. This is put in here because the principle of the training of a neural network and the logistic regression is not so different, really. I think there's too many formulas for you to keep paying attention for this. So I'm just going to keep it as what I was saying before. You're just going to be changing the weights and the biases so that you're obtaining the most discriminant, you're obtaining the best predictions out of it. Now we're going to go for the training. The training I was telling you before that you want to train all the weights so that every time you get closer to the best predictions, which would be found here. That's exactly what you're doing. I think if in structural you have already been checking, I think with the umbrella sampling, how close you were getting. This is like a pretty similar representation of it. So Basis agrees, I don't, of course. Yeah, it's not umbrella, it's kind of like that, but well. No, oh yeah, I see how that can be confusing. Never mind, this is not umbrella. Basically it's showing the marble going down, getting to the local minima, well, Yeah, or global. You would like global, of course. But at the end, you are just training simply by changing this weight I was telling you before. So, problems that can happen here. Overfitting. We all know how terrifying that can be, right? So, how do you do that? You can just diff some data outside, and that way you know what you're doing before, how well you're actually performing. But there's also another thing you can do, which is like try to generalize your model as much as possible. You can do that with regularization. You can do this by dropping out nodes. That's something that I'm not going to explain, but it's, the dropout is something that's very used in neural networks. There's ways in which you can prevent this, but at the end you want to have the data as general as possible in order to have good prediction. Now, neural networks and deep learning are, I would say, almost synonyms. That's not necessarily, but practically yes. So basically, deep neural networks, convolutional neural networks require a lot of data, and only recently we had enough computer power. There was like, thanks to gamers, we had like now very strong GPUs. Like that was the beginning of it, and we can start we can start accounting for a lot of data, very simple operations, very fast in GPUs. In the Bassett paper, I think they showed, I don't know if you, for those who read it, there was like a table with how much it took to train Bassett for a new setup of databases, of cell lines, if you were using GPU or CPU. And I think with GPU you were going, I think 20 or 30 times faster than with CPUs. So that was, of course, it was a very good GPU also. But that's something that has happened before. Some neural networks that now take a week to train, before you would need a whole year or even more to do it. So that is like what we're talking about deep. It's like you've got a lot of data and you can therefore extract more complex behavior. You can make larger neural networks. You can put more nodes, more everything in there. But that's not the only reason, of course, that we've got more data and that you probably was saying, but like we also have better algorithms that are making the training better. We talked about the ReLU activation function, which was something extremely good to prevent vanishing gradient. And right now, everybody can just download, for instance, TensorFlow, which I think probably all of you have heard of, and Keras, which is implemented, can be done from there. And in less than 100 lines, you have your neural network. You don't have to do everything manually as it was before. So thanks Google, thanks many other people, but yes, that's why deep learning has been advancing so much. Well, this is a bit what we were showing before, and for each neuron, here you can see the whole thing of it. You've got the summation and then the activation function for each of the neurons. You've got bias. In this case, I think they're putting it same one for each of them. But this is exactly what is happening in a neural network for each, but instead of only for one neuron, here you're seeing absolutely all together. For a very simple one, who has seen how deep neural networks look? Like a representation of it, yes? Can you see even the lines connecting the neurons? Yes? Or do you just see like a mass of things just getting together, if it's fully connected?
You mean like a deep dream or something? Like where they zoom into a neural net and show the representation?
Yeah.
Yeah. In the beginning you see a lot of lines, but if you go even deeper you see shapes of like eyes and...
Oh, no, I'm talking only about the connections between neurons. If you're having like 100, then another 100, you're having another 200, eventually it's just a mess. So this is like only 3 and 3, 2 hidden layers. If you start looking at how complex some of them can be, you might be finding instead of 3, you've got 100, you've got 100, and then you've got like this 100, this one connected with the other 100, the other one connected with the other ones. Eventually, this becomes large. But the principles for each neuron are exactly the same. You've got the addition of the products and the activation of the, so and then you put that into the activation function and you obtain output, which is set for the next one. Yeah, this is exactly the same that was done in Bassett. And I'm going to skip this because I think we have already understood it in the previous one. Is anyone who doesn't understand the convolution step? Take that as a no. And we were talking about how to convolute with 1D. Exactly the same thing. You've got the one, well, here it doesn't show the one hop encoder, but at the end what you're having is like only convolution in one direction, and then you, what do the filters represent? I think we can leave that for later, for when we interpret thing actual button. Pulling, max pulling, this is already done. What the effects, yeah, what effects does it have on the data? Who wants to tell me that? Pulling. We shot, yeah?
It's more like a question where I don't really know how to answer it. What effect does it have?
The pulling?
Yeah.
Okay, well let's see if someone can say it. Otherwise I'll do it. So do you remember the signs? You had a filter and you were getting something out of that, and then you were pulling, yeah? Well, that's an effect of it.
Lower resolution.
Sorry?
Resolution lower.
Resolution, yeah, okay. But like, what happens with the information you're passing to the next step?
It's a bit more generic, right?
It's more generic,
Yeah.
but it's still representative, right?
Mm-hmm.
Or should be at least. We have seen how the line can disappear. At least it should be there. Okay, well, you have already mentioned the dimensions. And well, this is basically how big you make the filter, the pulling.
Yeah.
What isn't clear?
My question is, okay, you're reducing, and then what is it that you do with the reduced set? Like, you're using it as a predictor?
No, the reduced set is just, well, the reduced matrix is what you're continuing with, basically. So from this, which is 6 by 6, if you're making pullings of 2 by 2, you're going to end up with a 3 by 3 matrix, for which you only have the positive values, so that the maximum values of each of these squares here. So you're expecting that only keeping the maximum values is going to be enough to represent what was here in a smaller dimension. And this also helps with generalization because you're not looking so much into the details anymore. Is it at least intuitively?
Sure.
Yeah?
Yeah.
Okay, good. That's what you were expecting, of course. Well, we talked already about the activation functions. I don't know if everybody got the activation functions because there was like, I didn't explain a lot of the leaky, well, there's no leaky relief here. But I think I mentioned already there's no vanishing gradients. This basically means like it's better for training because vanishing gradient can slow down your grading, so your training. Grading is also slow, but that's another story. Yeah. Okay, but this basically are, in each of the neurons, you're using one of this. In different layers, you can have different ones. You don't have to stick to the same one in every one. There's different architectures, and you can be creative and create your own one. Each layer can have different ones. Have you seen it also by neuron difference? Because I have not. Usually they put by layer. So if you've got 100 neurons in a layer, I think they usually have all the same activation function.
Yes.
Usually.
Between layers, you sometimes switch.
Yeah, between layers you change, absolutely. So what I'm trying to say is like this, so if you choose ReLU, that doesn't mean that you're keeping it for the whole network. You might be getting this for the first layer, and then you decide to go for sigmoid for the following ones, or for, well, sigmoid is very nice for the last step, so you've got an actual probability between 0 and 1. Yes, more of this, more convolution. I think we have already had a fair share of this, and we don't have to go more into it. This is going again for it. This is exactly what we're saying, like convolution, then you can pull it and get reduced, you do more convolutions, gets larger, gets reduced. And the same for DNA. So basically, what you're doing for images, because most of the 99% of the examples you're going to find on the internet are about images. It's all because it's simpler to see. You can use it with any dimension, even with 4 dimensions, 7 dimensions. It would be hard to visualize, or impossible maybe, but you could do it anyway. But yeah, cost function. Do you remember what the cost function was? Because this is about the training now. Before I defined the training, as the reduction of the cost function. And you could do that by changing the weights or changing the biases. This is pretty much the same, just it's, I think this slide is highlighting how this can be expensive, and you make the use of mini batches. You already have a representation of that, which you could go back to later. There's many tricks that have been done to accelerate the training. You've got different kinds of gradient descent. Here, I don't know them all, by the way. I don't know if anyone knows them all. I was told that there's like whole theses written about how to make this faster. So let's not step into this. Just for you to know, it's way beyond gradient descent and stochastic gradient descent, which I need here. And I think this should be an animation. It's not there. So now we're going back to Bicep. Okay, this is, we have already seen it. You've got all the dimensions clear, but now we're going to go for this. What is this? Who has the paper? Sir? Sorry, I can't hear you. Yes? Sir? Yes. And what is it representing? It's representing the accessibility. Oh, sorry. You.
No, go ahead.
No, you start. Come on. I start. You continue. Come on.
Yeah, accessibility per cell line.
So the cell line is this.
Yeah.
And this is what?
That's whether or not that.
Yeah.
That's the hypersensitivity site. Yeah. So if it's dark, that means it is
okay, so
accessible.
I think there were like 2,000 hypersensitivities sites. I'm not sure, but it doesn't matter. One more? 3? 20 more?
Let's say 22,000.
See?
Go ahead. Okay, fair. 2 million. Okay. So yeah, basically this is for each cell line. It's showing the hypersensitivity site. This, what is this? No, this is not a rock build itself, right? Well, it's the area under the curve of that. But like as itself, like what is this plot? What is it? Why do they put it there?
Comparison between SVM and Bassett.
Okay, so they are basically showing off, right?
Yeah.
Yeah.
Yeah, that's it.
Yeah.
They
want to see how good they are.
SVM, yeah.
So that's it. No, good. I want you to be critical of this. That's good to know. So what is this?
That's it.
So what is this? Why did they put it there?
For each.
I should have read the paper. It's a cool one.
It's the rates per. Is that cell lines?
Yep.
Yeah. Yeah. Those are the cell lines, and how well it predicts each cell line.
Yeah, but well, if they just show me here, like, oh yeah, nice plot. But
yeah.
Yes.
That's it.
Maybe like you've got different cell lines and well, the performance on the different cell lines.
But they are using it for the same method. They are using Bassett, right? So they are not here showing how good they are compared. They don't care about how good they show if a cell line is predicted better than the other. They are trying to show you something. Here. Shall I give it away already? I should have brought chocolate just for making people. Never mind. So, if I tell you what happens here, what are the values in this vertical line, like in point 2, for instance. If I put this figure in a paper, I'm telling, look at how good I can predict before it goes wrong. Look at how good my best predictions are and how confident I am of my predictions being good. That's pretty much it. It's more showing off. That's good. Just put it in your paper. You want to be published, you want to be in Nature, right? Put it there.
There is more to that, Paul.
Please be my guest.
This also visualizes that not every cell line is equally straightforward to predict accessibility for. So these are actually chosen to represent different quantiles. So if you rank the cell lines based on how well your predictions of accessibility are for that cell line, which is basically slicing rows across your final matrix of predictions, then you see at the very top the predicted accessibility for one hypersensitivity site in however many columns there are worth of cell lines. And then below that you see in which of those cell lines it was actually accessible. So for some cell lines the prediction will always be closer to the actual value than for other cell lines. And this AUC plot, or this rock plot in the bottom, shows for 5 representative cell lines of their quantiles how many you get right before you start making more mistakes.
Which is good info also. Let's see.
So back to my question. So the prediction and the actual, they're both kind of
Prediction?
like what are those?
Oh yeah.
Yeah.
They're what? Sorry.
Like what kinds of data are they?
So, well, for the actual, you get it from, I think they go from experimental data, if I remember correctly.
It's the heat map.
Yeah, but
It's one row out of the heat map that we started with.
Okay.
But that's from experimental data. I think that was the question, right? No, I thought that was a question. So yeah, it's an actual gold standard. So yeah, it's really just normal how you did it in the fundamentals course, for instance. Yeah. Okay. What is this introduction? Introduction of a database that contains the binding motifs. I think it's a good resource to have. This also plays a role because later we're going to be showing how many of the filters that were being used were already described here. Yes, this is the plot that I was talking about. This is the plot that I like. Somebody explain it to me. Come on. It's so not going in the direction I wanted to be. Guys, read. Yeah.
This is a tricky plot though.
Yeah, it
for
is.
People that have read the paper.
It is, absolutely.
Maybe start simple. Can anyone that has read the paper explain the colors of the dots in this plot? Can anyone that has not read the paper explain the colors?
We've got a reader.
Whether the found motifs are annotated?
Yeah.
So, what annotated? What does it mean?
Yeah. That's
Clear found in the database would be important.
Okay. And the other ones are not. Okay. So, why is this important? Yeah?
Motives, filters with motives that have a very large influence on correctly predicting accessibility were often not found to be annotated.
Yeah. So, what would be the direct application of this?
To find new.
Give candidates a place, right?
Yeah.
That'll be a very good one. Is there something deeper in this plot you would like to say?
The x-axis.
Yeah, that's what I want.
Oh yeah, I didn't put the formula anywhere. Is it?
Even without the formula. Can anyone hazard a guess what information content is? Does the term low complexity mean anything to you?
Yeah, that's...
We've discussed this before and how it makes, for instance, short read sequencing very difficult to map.
My God, H, entropy.
Anyone?
The entropy per that particular region,
Yes.
and the complexity of it.
Yeah, so this is very closely related to entropy, exactly. The sequence motif logos that we've seen before, the height of the bars is actually expressed in bits of entropy. So having a very high bar means that it is very important that that specific position is that specific nucleotide. Meaning that if you have a lot of specific combinations of nucleotides that must co-occur to map to this filter has a high information content. Because there is only a few places in the genome where you could find this sequence by chance.
Yeah.
Inversely, if there are many different sequences that would result in a high score for this filter on a certain position, so if the, say, the product of the heights of the bars is relatively low, that is not the exact equation, but you get the idea, then this specific filter does not have such a high information content because the motif is, shall we say, promiscuous or ambiguous.
Mm-hmm.
But what this plot shows actually is that there is an unexpected relationship between how big an influence the presence of a motif has on the final output of your classifier and the information content of that motif.
Does this indirectly also imply that the size of the motif is important? Because the smaller the size, the less the chance you have that it may have.
Yeah.
Yes.
So it's very small bits that actually have a huge, bigger influence.
Yeah, well, there's also many genes that you have to be expressing at some point, right?
Yeah. And can we also select on those points and see what they are and try looking for them in the genome?
Visualized. Like you see in the figure itself, there is 3 sequence logos plotted. And you indeed see that the lower bars have lower information content.
Shall I try to make it bigger?
And then finally there is one small interesting point of discussion that you can see in this figure, and that is it very strongly visualizes the publication bias. The motifs that are annotated, so the ones we already knew about, are the ones with high information content. So older technologies or more primitive ways of searching for these motifs have only resulted in a hit if the sequence that you find is very specific. So the statistical power of low information content motifs is lower, so the amount of data that we have looked at to find them before was not sufficient.
Okay.
Yeah.
Yeah.
So, well, shall I keep asking what this is? Because not many people haven't read it. Yes.
I have a question about the previous plot.
Yes.
I think it's strange that the very influential motifs have very low information content.
Yep. That's exactly
Is that
what's
due to the bias, or is it just if you remark it and it's like, okay, you can see that, and that's it?
No, the bias is different. It's not.
The bias is the distribution of orange and green plots along the x-axis.
But how is it done, or is that not really an explanation?
So,
If you say that the motif is ATG, I can think of at least 22,000 times that appearing on the genome. Do you think that's something that a reviewer would be happy to see? Like, oh, yeah, this guy is maybe probably just making it up. That's the way I'm seeing it, at least. If you're showing something that's very low complexity, people tend to not trust it. Or different view?
I'm thinking of how to phrase this correctly. There was a very interesting keynote talk at BioSB on the Tuesday session this year, showing that the highest affinity binding sites of transcription factors are not the only functional ones. In fact, these transcription factors can still bind the DNA that is only 1,000th, so 1 per mil, of the affinity to its cognate motif, or at least what we think is its cognate motif, and still have a functional impact on gene expression. Less than 1,000th of the best affinity in the genome, then things become less functional and more, say, sporadic. But this shows that the way we think about cognate motifs and what is the motif of a transcription factor binding site, is perhaps too rigid. And that even less specific binding sites are indeed very functional, despite being more promiscuous throughout the genome. Because this is only the first layer of filters, right? This specific method uses 3 layers of convolution. So the very first layer captures these sequence motifs, and that is exactly what they've extracted and plotted here. Then this is rectified and max pooled in a step of 3. And this new matrix that you've now generated becomes the input for the next layer of convolution. So there you start looking for combinations of these motifs in their presence. And then the final convolution layer starts looking for combinations of combinations, and that becomes the input for a neural network that classifies whether or not you are accessible in a certain cell line. So that goes to show that even low complexity or low information content sequences can be very critical if they are combined with other low or high information content motifs. Does that answer your question, Alice? And I think that is all we have to say about this figure.
Yeah, I think so. I think so. No, like I think it probably takes a bit more work from your side to understand that neural network. Well, like the convolutions in this case, the first one are pretty easy to interpret just directly with filters. But even past the point of filters, when you start looking at different layers of neurons, the earliest layers are capturing more direct effects, like the most general effects, and then it gets even smaller. So like this combination that Bob was talking about can get even further. So like with the first one, maybe you're getting, oh, there's like this very simple one, but this very simple one combination with maybe a strong one, or 1,000, like I'd say 20 more simple ones are making a very strong effect. So yeah, I'm gonna say it anyway. What is this? No?
Keeps compared.
Yeah. Compare what with what?
Their
What-
results and baskets.
So what is this thing on top? What for each of them? So that's the actual motif we have in the database, right? So what is this thing below here?
That's it.
Not prediction.
Or result. Suggestion.
It's the filter. It's one of the filters that was used. Yes. And like these are the ones that were shown to that were shown to be important, to be relevant, that Bassett was using, and they were already present in the database. You can see that they are not exactly the same, but I mean, if you don't see the similarity of these two, for instance, then so you can already see that simply by letting the network extract everything, you can already
Do you think that maybe the database is incomplete and the neural network is correct?
the database.
Yeah, the database did the top one is the motive that showed is from the database, right?
Yeah.
And those are all the ones they found in the database.
Yeah.
And the below one is found by the neural net, right?
Yeah.
Trained on all the sequences, right?
All the sequences that they used, yes.
Yes. So could it be that the top one is maybe incorrect because that's human manually selected probably?
Well, I guess we would have to look into how the database was done, but I doubt it.
No.
Like, I think they are pretty they're probably experimentally checked already. Like,
So that's actually how we found these. For these transcription factors, we have found, for instance, through ChIP-seq or other co-precipitation sequencing experiments, these transcription factors are known to bind to a set of sequences. And then by aligning those sequences and compiling a profile, like you would have with sidebars, for example, you can tell that in this position we observe an A, like an 80% of all the sequences that this transcription factor binds to.
who doesn't know what ChIP-seq is? And that's okay.
What is
ChIP-seq.
ChIP-seq?
Chip sequencing. Chromatin immunoprecipitation.
Oh, no.
No?
I
Okay.
thought he said cheesesteak.
Maybe that's...
It doesn't really matter.
doesn't matter.
But basically what we have is a database of sequences and what transcription factors they bind to. Or rather, conversely, for transcription factors which sequences they bind to. So this doesn't have to be complete, right? But these logos that you see at the top are simply a summary of the sequences that we have found bound to a certain transcription factor.
Yeah. So, yeah, that's actually correct. So, that's experimentally determined, and you can... yes, you can observe that depending on the cell line, you're finding different filters having different importance. This is because of the sequences being slightly different due to mutations. And I wanted to ask what they did with this, but no one read it. I'm sad, guys. No, okay, so in silico mutagenesis, let's not even go with what they did. Just looking at the name, what does it mean?
That they just change the sequence.
Yes. Yes, you just change it in the computer. That's it. Who can tell me what they did here? A wild guess. Okay, let's say you've got, so this is only one of the sequences, right? So this is only, yeah?
For every of the letters on the top, they change it into any one of the three others, and then they check the difference in the relevancy.
Yes, I don't remember if it was, yeah, this was the actual letter. Yes, that's the letter that was there. Yes, that's exactly it. They changed for each position the letter that was originally there by the other three, and they checked how this change had an influence on the accessibility of the site. And then, yeah, okay. So what is this? I'm so loving this. This is coming directly, well not directly, pretty directly from the previous plot. So from here, you're having ups and downs, right? Depending on the accessibility, you've got loss and gain, and the influence that it has if you are first mutating here. What happens here? Yeah, maybe that's better. Yeah, what happens here? This point here, that's a nice one. What happens here that is not happening here?
Loss of accessibility.
Yeah.
Yeah.
So you're losing a function there. You're losing a function
Mundo.
by mutating there. If you're looking at this and you like,
obviously that's a site.
that's a place that has an importance in the.
Yeah.
Yes.
Yeah.
Good. Good.
So before you go to the next slide,
Yeah.
we explicitly discuss what is visualized in the bottom two tracks of this figure.
Okay. People.
In the what? In the bottom? The bottom?
No, well, I guess this one is already pretty interpretable from just the numbers
So
there.
for the people in the back, the legend says the blue line is losses and the red line is gains, and the green bars in the bottom are annotated philo P. Someone tell me what that means.
The final P or something.
No? Sorry?
It's a method, but it tells you something. What does the final P score signify?
Conservation.
Very good. And what do we know about conservation? Yes, things that are well conserved are likely to have an important function. So, do we understand then why this FOSS motif that has been identified, the TGASTCA motif,
Yes.
does that have a higher or lower conservation than the area around it?
High.
A bit higher.
What then would you expect from the red and blue lines in the graph above it?
You should change it to... I just said it.
Would you expect a high red there? I see no. Why not?
Well, if it's less accessible, then that means there's something there.
So if it's more accessible when you make mutagenesis, you mean?
Yeah. Yeah, if you mutate it, then the function is lost, and obviously it has a
No,
function.
The function is lost if you've got a high blue line. If you've got a high red, you're
Yeah.
actually increasing it, right?
Yeah.
So do you think that would be very conserved? I'm actually.
No, it would. No.
It could be also.
When?
So
No.
one way of
If it's not-
interpreting this is that because this motif has a function, it is conserved, and if this motif is perturbed, then, specifically in this case, you would lose accessibility.
Yeah.
Right? Because the red and blue lines are the sums of the red and blue cells in the matrix above.
But then you assume that if something is really conserved, it's also accessible.
Can also be the
Not
other way
necessarily.
around.
Yeah, that's why I said
Exactly.
maybe not. Yep.
So I do not recall by heart if this is across all cell lines or for a specific one.
Maybe
But the idea is that you can estimate now, with your model, that every time you change a nucleotide into one of the three other options, what does this do to the accessibility of this site? And that is what this heat map shows. Right? And if you add up all those gains and losses of accessibility for your three mutants, you can plot the blue and green lines. And we would expect that a big difference in accessibility would coincide with conservation, which is why they've plotted the final p-scores beneath them. And then finally, because we don't want to watch individual nucleotides or individual hypersensitivity sites one by one, but we want to summarize all this information together, we go to the next slide. Knowing what we know now, after the discussion of the previous figure,